Welcome to Biology Made Easy. Biology Made Easy are biology lessons designed for high school and secondary school biology students. You will find the lessons very, very good for yourself and for higher learning. In our first lesson, we introduced biology. We also discussed the characteristics of living things, since biology is all about living things, organisms. And you had assignments to do. There were 2 assignments that you were required to work on. So all the life processes go on in the cell, and there are organelles, various organelles that do that. So therefore, we are looking at biological techniques. We are going to look at what a specimen is, sectioning. We are also looking at symmetry, and then orientation. Right. Objectives for today. We hope by the end of the lesson, students, you should be able to list biological skills and biological attitudes, the skills and attitudes that every scientist and a biologist should possess or should build. Students should also be able to explain the terms specimen, sectioning, orientation, and symmetry. Then you should also be able to find the differences between longitudinal section and then transverse section and vertical section, and then explain the importance of these different sections, and then importance of symmetry and importance of orientation. Good. Now let's go on. Biological skills. What do you think every biologist or scientist, the skills that a scientist or a biologist should have? Enumerate some. Think about some. Well, if you are a scientist, what do you think? You should be very observant, all right? So there's the skill of observing to see things. You should be able to be measuring things, planning your work, all right? Recording, you should be able to record meticulously. Classifying things, analyzing, drawing, making models, making inferences or conclusions. These are some of the skills, all right? Well, is skill different from attitude? I think if you are a scientist, you should possess some kind of attitude, all right? Do you think there's any difference between skill and attitudes? Well, I think they are not too different, but look at it. You have a skill of observing things. It means in your attitude, you should be very observant. You have an observant attitude. You have a questioning mind. Why is this like this? Why is this like this? You should be diligent. You should be persevering. You are looking for something in a specimen. You can't find it. You don't give it up. You persevere. So one of the attitudes is a persevering attitude. You should also be able to organize things well, all right? These are some of the attitudes. Now, without wasting time, let's look at what a specimen is, because that's one of our tasks for today, to find out what a specimen. Scientists talk about specimen all the time. What is a specimen? Well, a specimen can be a whole organism like this, or part of an organism. So plant can be a specimen, or part of an organism that's just a leaf can be a specimen. And even so, if we are taking just a tiny bit like this to put under the microscope, 1 cell thick, it's a specimen. So that's simply what a specimen is. For your assignment, you are required to find out when is it necessary to use the whole organism as a specimen or just part of organism as a specimen. Good. So that's about specimens. Now let's go on to look at sectioning. What is sectioning? Sectioning is cutting a specimen with a specific aim in mind. Cutting a specimen with a specific aim in mind. What are the reasons for sectioning? What are the reasons for sectioning? You have any reasons for sectioning? One is to see the internal structure of the specimen. All right? To see the internal structure. Another thing is to get very tiny pieces like we did, very tiny pieces that we can put into a microscope. You want to see the internal structure, you have to section. You want to get just a tiny piece to work on, you have to section. These are the reasons for sectioning. Now let's look at the types of sectioning that there are. You have longitudinal section, transverse section, and vertical section. Now let's look at longitudinal section. It's a cut that runs the whole length or part of the length of a specimen. For instance, this carrot, this is the length. All right, that is the length. This is the breadth. If a cut is made along this length, then what we get is longitudinal section. As you can see, this orange, this is the length. A cut along this length gives you longitudinal section. We can define the transverse section as a cut across the length. This is the length, a cut across. So you have neatly transverse section of the orange. All right? Good. The vertical section is done in structures that doesn't have any specified length or breadth. That's the vertical section. For instance, skin, it's difficult to find out what is the length and the breadth. And so you have a vertical section of skin. Right. Now, let's look at the different sections of the fruits, orange fruits. You have the transverse section. You have the longitudinal section. Right. Let's look at the differences between the transverse section and the longitudinal section, TS and LS. You look at the TS and you look at the LS. You see that the LX has a long placenta. The TS have a small one stuck in there. It's the same fruit. All right? That is it. So there's difference between the TS and the LS of the same specimen. What importance does this show you? Look at them again. It shows that different sections of the same specimen show different things about the same specimen. Look at the specimen again. You see that the LS has only 2 locules. The TS has a lot of locules of the same fruit. So different sections show different things about the same specimen. Then also, different sections of the same specimen can be done so that you have a good picture of the same specimen. Good. So that's about sectioning. We'll look at sectioning when we look at microscopes and see how sections in microscopes should be done. So now let's look at symmetry. You've heard the term symmetry. What is symmetry? A very important term in biological techniques. Let's define symmetry. Symmetry is a line of division that bears in mind proportions and which cuts a body or specimen into equal parts. Look at it. A line of division that bears proportions in mind and cuts a body into equal parts. It means before cutting a specimen, you should look at the symmetry of the specimen. So, symmetry is important to note before cutting. What are the types of symmetry there are? We have bilateral symmetry and radial symmetry. There's also asymmetry that we have to note. And note that symmetry, the cutting is made in such that the halves that are gotten should be proportional. For instance, in bilateral symmetry, the organism is made in such a way that there's only one line, one plane, one line that the cut should be made, that the two halves will be proportional and equal and mirror images. So here you see that the cut is made in such a way that you can only, it's only this line, that this area and this area are the same. So this is bilateral symmetrical, only one plane. You look at the zebra is also bilateral symmetrical. You see the ears, bilateral symmetrical. And then you see this animal too is bilaterally symmetrical. Look at its limbs and look at, you can quickly know that there's a place that you can cut and you can have the left and the right proportional and mirror images. So that is it. Then we have radial symmetry. In radial symmetry, the organism is such that you can make a cut in many, many planes and have mirror images. We can cut this organism in this plane, and this half will be proportions and mirror images. We can also have a cut along this plane, and A and B will be the same. Here too, A and B is the same. So in radial symmetrical, organisms that are radially symmetrical are normally circular or tubular. And any cut along the diameter will produce mirror images. Any cut along the diameter. So if there's a center here, any cuts through the center from one end to one end will produce mirror images. So that is radial symmetrical organisms. In any case, what's a diameter? Let's define what a diameter is. A diameter is a line going through from one side to another through the circle. You have a circle, like I have a circle here. The center is here. Diameter is a line going through the center to one end here, another end here. So I can have a lot of diameters. If I can go through this way, I'll still have this part and this part being proportional and mirror images. So organisms that are tubular or circular are radially symmetrical. There are so many planes along which the organism can be cut, and the proportions will be the same. I can cut from here to here, and this plane and this plane, this part and this part will be mirror images. These organisms are radially symmetrical organisms, and the cnidarians are the organisms. All the cnidarians are radially symmetrical. As you can see, this organism, Aurelia, you see how it is? This one too is radially symmetrical. So what is the importance of taking note of the symmetry before cutting the organism? It's very, very important. Well, it's important to take into account the symmetry because what you see depends on the plane along which the cuts are made. Different planes will reveal different things. For instance, if this man is cut along this plane where it's bilaterally symmetrical, we should be able to see organs that are along the plane. If a cut is made, if the cut is made along a different plane or not through the symmetrical line, let's say a cut is made around through here, we will not be able to see these structures. So what you see depends upon the plane you cut. So the symmetry is very, very important before cutting is done, sectioning is done. And the symmetry is also very, very important before sectioning because you can decide to cut along the plane for which you cut, and you only study half of the organism. For instance, if we cut bilaterally, we have this plane, we have this, half of this will reveal everything that is in the other half. Radial symmetrical organisms do the same. If we cut we cut along the right place, we get what is in this plane is also what is here. So once we know what is here, we know what is in the other side. And if we don't cut along the right plane, we don't see what we should see. If instead of cutting along this plane, we cut around here, which is not through the diameter, we may not see these structures. We cut here, we won't see these structures here. But if we cut along the plane, we get to see what we want to see. So knowing the symmetry of the organism before sectioning is very, very important. So now let's get to orientation. That's also another thing we have to look at before our lesson closes for today. So what is orientation? Orientation describes how the body or part of the body is positioned with respect to the surrounding environment. How the body is positioned with respect to the surrounding environment. For instance, orientation looks at its positioning, all right, with respect to the environment. So this organism, this part is its dorsal view. That's the back view. This part, we're looking at this organism from this end, we are looking at the anterior view. If we are looking at this organism from this end of the environment, we are looking at the posterior view. Looking at the organism from underneath is here, we are looking at the ventral view. Ventral. If you are looking at the organism from its side, we are looking at the lateral view. So that is orientation. Orientation, one may ask, why do we have to worry ourselves thinking about orientation? Very, very important. If you are describing an organism like this, you need to state the orientation. It is at the anterior end of this organism. That's the anterior end. You are looking at the organism from the head area. Anterior end is the head area. So at the anterior end, there's a head there. We have an eye, etc., etc. If you are looking at this dorsal view, you are looking at it from the back. You are looking at the ventral view, you are looking at undersurface, has this and this and this. Orientation is very, very important when you are describing a structure. If you have this fish to describe, this fish has this spine on its mid-dorsal view. So we can say there's a spine on the mid-dorsal view, or there's a spine on its dorsal area. It also has 1, 2, 3, 4, 5, 1, 2, 3, 4. It has 4 bands, dark bands on its lateral side. So orientation is very important when you are describing. If a surgeon has to describe a structure, he need to state, all right, this structure is found on the dorsal surface, and very, very, very specifics. It will state the area and also draw. So orientation, very, very important in describing. And then orientation, sometimes one will be told to draw the anterior view. If you have to draw the anterior view, you know you are drawing the head. Draw the lateral view, you are drawing the view from the side. That is orientation. Very, very important to note. Now, this brings us to the end of our lesson for today. There's some homework or assignments, and these are the assignments. 1. When is it necessary to have the whole organism as a specimen? That is 1A. When is it necessary to have the whole organism as a specimen? 1B. When is it necessary to have parts of this organism as a specimen? 2. Look at the following pictures or organisms. Specimen A, B, C. State whether the specimen is bilaterally symmetrical or radially symmetrical. 3A. What is the shape of specimen D? B. Describe the anterior view of specimen D. that is the end of the lesson for today. Goodbye. Thank you.